Swayam Prabha. Digital India. Educated India.
Hello learners. So today we are going to discuss about the topic variations from the paper Evolutionary Biology. Now this is the fourth module in the curriculum of the paper Evolutionary Biology. We will cover this topic under two lectures. This is the first lecture where we will discuss how variations act as a raw material for evolution. We will also discuss what is the connection between variation and evolution. Or in other words, why variation makes an important part whenever we talk about evolutionary biology. I am Dr. Sudhir Verma, working as Assistant Professor in Department of Zoology, Deen Dayal Upadhyay College, University of Delhi. In this lecture, we are going to learn about variations. Particularly, we will restrict our discussions to genetic variations or heritable variations. As we'll move ahead in the lecture, you will see there can be different kinds of variations. But wherever we use the term variation, we actually mean the genetic or heritable variation with respect to evolution. Then we will see why it is important to study about variations. Why is it an important part of evolution? We'll also discuss the different kinds of variation that does exist between the individuals and the population as a whole. And lastly, we will talk about the various source or causes of variation between the population and the individual as such. Moving ahead, let us first try to understand what do we mean by the variation or the genetic variations. All sorts of differences which we observe between individuals of same population or a species, and even the differences that do exist between offsprings of the same parents come under genetic variations. In other words, whenever we use the term variations or genetic variation, we are talking about variations that do exist in the alleles or the genes both within and among the different populations. So whatever differences that do exist in terms of allele or gene between individuals which are belonging to the same parents or which are belonging to the same population or species, they come under variations. Now, these genetic variations is different from what we call as environmental variations. If you define genetic variation, it is the variation of genomes between individuals in the same species. Whereas the environmental variation is the alteration of phenotype of a particular genotype as a response to the environment. Here, the examples can be the coat color of animals, the skin color, color of hair, color of eyes, freckles, dimples. All these are the genetic variations. Whereas the phenotypic differences which do exist in the identical twins can be taken as an example of environmental variations. Though these twins do have same genetic position or genotypes, they do show the phenotypic variations which do exist because of environmental variations. The genetic variations can be caused by mutation of genes, gene flow, random mating, random fertilization, crossing over, and so on. Whereas the environmental variations can be caused mainly by the external environmental factors. These can be the temperature, the light, moisture, climate, and so on. The genetic variations do pass through generations and through natural selection, and thus they are an important part or factor in evolution. whereas the environmental variations do not cause changes in the genome. Hence, they have no effect on the evolution. So, since the environmental variation are not going to have any impact on evolution, hereafter, whatever we discuss, we'll discuss about the genetic variation, which do have a role to play in the evolution. The evolutionary potential and the genetic variations are nothing but the two sides of the same coin. And why we are saying so is because without variations, there would be no evolutionary change. Why we are saying so is, if there is no variations, just assume a population where everybody is same. So where will natural selection operate on? Where will evolution will take place? In this kind of situation, there can be no evolution or no natural selection as such. In other words, if you see the contradictory situations, without evolution, variations would be unnecessary. If there is no evolution which has to take place, there is no requirement of variations in that case. So that is why we call that evolution and variation are nothing but the two sides of the same coin. Now, let us move ahead to discuss various types of variations. Variations can be classified according to different criteria. Now, depending on which parameter we are selecting, there can be different types of variations. For example, if we are talking about the affected trait, we can differentiate between morphological variation, physiological variation, cytological or behavioral, and so on. So there can be different type of variation depending on the traits which are being affected. If the parameter of study is the impact of variation, the variations can be useful, they can be harmful, or they can be neutral in its impact on the individual. If you are studying parts in terms of variation, then there can be meristic variation or there can be substantive variations. Depending on the degree, they can be continuous variation or discontinuous variations. Depending on the cells which are being affected by variations or the changes, there can be a somatic variation in contrast to germinal variation. Then further, if you see the cause and effect kind of relationship, there can be genotypic variations which are constitutional in nature, or there can be the phenotypic variations which are nothing but the observable changes. Then you can also have determinate or indeterminate variation depending on whether the direction of evolution is fixed or not fixed as such. So depending on the parameter, we can discuss different kinds of variations. Now let us quickly see what do we mean by this. Let us first talk about somatic versus germline variations. That means the classification of variation based on the type of cells which are being affected. Now, if you see the character and try to differentiate between somatic variation and the germinal variations, there can be, depending on the cells involved. In somatic variations, or which you also call as somatogenic variations, in these only the somatic cells are affected. Whereas in the blastogenic or germline or germinal variations, it is the germ cells of gonad which gets affected. If you talk about the period of origin, somatic variations do occur during an individual's own lifespan, whereas the germinal variations occur during gametogenesis in the parent, and that is why from the gametes they move on to the next generation. If you talk about the factors inducing variations, in somatic variations, they are developed in response to changes in environmental factors like temperature, food, humidity, light intensity, etc. Whereas the germinal variations are developed either due to the mutations or recombination of genes or maybe other reasons or the causes. In terms of role in heredity, the somatic variations are non-inheritable. They are not getting passed on to the next generation. Whereas germinal variations are being passed on to the next generation and hence they are inheritable. The examples may be like better developed muscles in an athlete, which has been acquired in one's own lifetime. But it's not necessary that its progenies are going to have the same developed muscles, because this is an acquired character and it is thus a somatic change. Whereas polydactyly in man, which is a genetic character, they can be passed on to the next generation and hence it shows the germinal variation. What do we mean by the meristic versus substantive variation is depending on the change that is involved. In meristic variations, there is an occurrence in the number of some body parts. For example, polydactyly in man, that is an additional digit in man, which is a numerical, a numeral change. Change in the number of segments in earthworm, this is also a meristic variation. Whereas, in contrast, if you see the substantive variation, they do occur in terms of shape, size or color of the organism. For example, the brachydactyly. If there is a short digit in an individual's digits, this is an example of substantive variation. So there is no change in terms of number, but there is a change in terms of size here. Height of an organism is another example of substantive variation. Moving ahead, what do we mean by continuous versus discontinuous variations? In continuous variation, there is no distinct categories. There is no limit on the values and they tend to be quantitative. For example, if you take an example of height in human, there are no distinct categories. Can be, let's say, in the blood group of organisms. In blood groups, there are fixed categories. Either it will be an A blood group, B blood group, or AB blood group. But in terms of height of individuals, there cannot be a fixed height, 5 feet, 6 feet, and so on. There are variations among them. There are individuals which are 5 feet 1 inch, 5 feet 2 inches, 5 feet 3 inches, and so on. So there is no distinct categories. There is no limit on the values and they tend to be quantitative. Height, weight, heart rate, finger length, leaf length, these are all examples of continuous variations. Whereas in discontinuous variations, there are distinct categories, as I just mentioned, different blood groups. There is no in-between categories. There cannot be a blood group which is slightly O and slightly AB kind of thing. And these tend to be qualitative. For example, the tongue rolling. There is no fixed there is no variations in tongue rolling. Either you can or you cannot. Fingerprints, eye color, blood groups, these are all examples of discontinuous variations. Here you can see the graph representation. Continuous variations always show a bell-shaped kind of line graph. Whereas in case of discontinuous variation, you can always have the bar graphs. In continuous variation, there is a lot of genes and the environment which controls their expression. When I'm saying lot of genes, that means there is a polygenic inheritance. Whereas in discontinuous variation, there is a few genes. Limited number of phenotypes with no intermediates are there in case of discontinuous variations. Then there is a difference between genotypic versus phenotypic variations. When we say genotypic variations, that means we are talking about the variations which do exist in terms of genetic constitution. Whereas whenever we talk about phenotypic variations, there can be two factors which are playing role. One is the genotype, which is controlling that phenotype, and there is an additional factor called environment here, which can also control the phenotypic appearance of that character. So when we talk about genotypic variations, we are referring to genetic constitution. When we talk about phenotypic variation, we are talking about the differences or the variations which do exist in terms of physical appearance. Then we do have something called determinate versus indeterminate variations. For example, if you talk about the direction of evolution, If it is a determinate variations, there is a definite direction. There is mostly an adaptive direction in which the variations do exist. Whereas in case of indeterminate variations, there is no definite direction of evolution which can be predicted in which the variations are happening. For example, if you talk about Darwinian factors of natural selection, we do not know in which direction it is going to happen. So that is in case of indeterminate variations. Whereas we do not know the causes of determinate variations. Now, let us move ahead and discuss what can be the various sources of variations. See, broadly you can define or differentiate different parts of source of variations whenever we talk about. It can be the sources which are occurring at individual level, or there can be the variations or the causes of variations which are existing at the population level. So if you are talking about an variation which exists within the individual, there can be these many types. Whereas if you talk about population level variation, there can be a different set of reasons or causes of variations. Let us discuss them one by one. If we are talking about the individual level variations, we are going to talk about the chromosomal aberrations. That means the changes which are occurring at the level of chromosomes. They are further divided into the structural aberrations or the numerical aberrations. Structural means there is a change in the structure of the chromosome, whereas numerical aberrations can be the changes which are occurring in the number of the chromosome or the chromosome sets. Then there can be the gene or point mutations. That means the changes which are occurring in the nucleotides. Transposons also acting as a source of variation within the individual. Besides this, the genetic recombination in sexual reproduction, the gene regulation, they are all source of variation at the individual level. Whereas at the population level, the multiple allelism and the genetic polymorphism, continuous or the quantitative characteristics, and then there are different variation sources which change the gene pool, which may be the mutation as such, migration, genetic drift, or natural selection occurring in the population that can act as a source of variation. Let us try to understand these one by one, starting with the chromosomal aberration as a source of variation. Under this, we will discuss about the changes which are occurring in the chromosome structure and the changes which do exist in terms of number. The chromosomal mutations are nothing but the change in the number or structure of the chromosomes. If you see, we can easily differentiate or classify the chromosomal mutation into three different categories. These can be the chromosomal rearrangements, where there is a structural change in the chromosome. There can be aneuploidy, which is the numerical aberration, where there is a change in the number of chromosomes. And there can be polyploidy, which is also a numerical aberration, but here the whole set of chromosome is changed. So in contrast to aneuploidy, which is just a change in number of chromosome, polyploidy refers to change in the complete change in the set of the chromosomes. Let us talk about the chromosomal rearrangements first, which are nothing but the structural changes in the chromosomes. There are four basic types, which are duplication of the chromosome, deletion of the chromosome, inversion, and translocation of the. Let us quickly have a look with the simple representation. Let us assume that this is one chromosome and this is the centromere. So this is P and Q arm, you can assume here. What we mean by duplication is that the part of it has duplicated. So here the the B in itself has duplicated. So there is an additional segment B here which you can see. Now, if there is a doubling of part of chromosome, obviously it is going to alter the dosage of gene. This duplication can be of various types. First is the tandem duplication. Now what we mean by the tandem duplication is immediately or consecutively after the first one. For example, if it is ABC and this dot is representing centromere here and then DEF. Let us say the BC part has duplicated here. So ABC, BC, that means it is tandemly after this one. So it has duplicated and joined tandemly. This is called as tandem duplication. In displaced duplication, the duplicated BC part has joined somewhere else. So it is not immediately after BC, but it has joined somewhere else. In reverse duplication, the BC has duplicated, but it has reversed and now it has joined as CB instead of BC. So depending on how the duplicated part is joining the chromosome, there can be tandem duplication, displaced duplication, or the reverse duplication. The duplication can cause gene duplication and divergence. As in the previous lectures we have seen the globin gene family. The hemoglobin and myoglobin both are arising from the basic ancestral globin gene which has been duplicated and diverged. So this is an example of the effect of gene duplication or the chromosomal duplication. There can be sequence homology, the homologous sequences. For example, the hemoglobin and myoglobin in humans. This is a result of chromosomal duplication which shows the sequence homology. Then we have also seen in previous lectures the pseudogenes, which are nothing but the functionless members of duplicated gene families. These are all result of chromosomal duplication which play important role in evolution. Moving ahead, the next chromosomal aberration can be deletion. Here, as you can see, the B segment of this chromosome has been deleted. Obviously, if a part of chromosome is getting deleted, there is going to be an effect on the gene product. So, the deletion can cause imbalance in the amount of products. The recessive allele on undeleted chromosomes to express. So, there is a effect associated with deletion where the recessive allele on undeleted chromosome is to express. This is called as pseudodominance. Then there is an abnormality with respect to haplosufficient genes. These are all effect of the deletions. Then there can be inversion. Inversion is next category of structural aberration in the chromosome where there is a inversion or turn around 180 degrees of the chromosome. So this is the chromosome which has taken a turn of around 180 degrees Celsius, 180 degrees, because of which the ABCDEF segment now has changed as AEDCBF. This is another example. Suppose this is AB centromere, then CDEFG. After inversion, let's say it has become ABC and this DEF has inverted. That means rotated around 180 degrees and it has joined as FEB followed by G. It causes depression in observed recombination frequencies. There are two different types of inversion. It can be the paracentric inversion. Para here refers to next to. So it does not include the centromere. For example, in the same example which we see here, this is the centromere and the inversion is taking place only in this part of the chromosome. So it is not involving the centromere and hence it is called as paracentric inversion. Whereas in case of pericentric inversion, there is a involvement of centromere. For example, here if this is ADCBEFG, so they have involved the centromere here. This is called as pericentric inversion. Peri means around. The next category is translocation. In translocation, there is a movement of genetic material between non-homologous chromosomes or within the same chromosome. Remember, we are not talking about crossing over here where there is a homologous chromosomal involvement. We are here talking about non-homologous chromosomes or the translocation of genetic material within the same chromosome. It can disrupt gene function. For example, the position effect, which you might have read in your genetics classes. This is an example of translocation, because of which some part which has translocated from one position to another one in the same chromosome or in the non-homologous chromosomes can show the position effect. So where it has gone, it depends whether it is a heterochromatin chromosome or the euchromatin chromosome. Accordingly, it will show the same effect. Again, translocation can be of two types. One is called as non-reciprocal translocation, where there is a movement of genetic material from one chromosome to another without any reciprocal exchange. So it is just like a one-way traffic. From one chromosome, one part getting translocated to another one, but from another chromosome, it is not coming to the first chromosome. There is no reciprocal exchange. Whereas there is a reciprocal translocation, which is a two-way exchange. Means there are two chromosomes involved. One part from one chromosome is going to another one, and from the other chromosome also some part is coming to the first one. So there is a reciprocal exchange. Then there is something called Robertsonian translocation. In Robertsonian translocation, let's see if there are two acrocentric chromosomes. So here this is one acrocentric chromosome and there is another acrocentric chromosome, and if there is a translocation taking place between two. So the result can be the larger arm of this chromosome is getting attached with the larger arm of this chromosome. So it gives rise to a large metacentric chromosome here, and the smaller parts or the arms of these two chromosomes join together to form a very small chromosome, which ultimately gets lost. So this is an example of Robertsonian translocation, which you might have read under familial Down syndrome in your genetics classes. Normal Down syndrome is trisomy 21, whereas the Robertsonian translocation also gives rise to Down syndrome type symptoms, but it is a familial Down syndrome which is occurring because of Robertsonian translocation, not because of trisomy 21. Now let us move ahead to next category of chromosomal aberration, which is the changes in the number of chromosomes, or in other words, which we call as aneuploidy.